Hello everyone, my name is Ankur and welcome to RJPT Simlab videos. In this video, we will study about the common laboratory techniques for blood withdrawal. At the end of this video, we will be able to learn about the preparation of these common laboratory techniques, the site for blood withdrawal, the procedure of these techniques, volume of blood correction from these techniques, and the post-procedure care. Now, at first, blood withdrawal. What is blood withdrawal? Blood withdrawal is the common laboratory technique used in the pharmacological research to collect blood samples from rats and mice for various analyses. This procedure must be performed with care to ensure the well being of the animals and the accuracy of the samples. The statement says that it is a very common laboratory technique which is used in the research of the pharmacological purposes to collect the blood samples from the animals such as rats and mice for their various analysis. And to perform these procedures, it should be performed with much care to ensure the well-being of the animals, its safety, and the accuracy of the blood samples collected. Now, let's begin with the video. Here we can see we have the five different techniques for the blood withdrawal. At first, we have the retroorbital blood withdrawal technique. Second, tail snip blood withdrawal technique. Third, tail blood withdrawal technique. Fourth, cardiac blood withdrawal technique. And fifth, posterior vena cava blood withdrawal technique. So now we will see the first technique, which is the retroorbital blood withdrawal technique. As we can see in this technique, we have our animal samples, which can be either mice or rat. They are stored in the storage cage or container, and then in this technique, we have to pick one of the mice or rat. We have to place them on the tray. We have to handle them very gently, and then with the help of capillary. This instrument that we can see is the capillary tube, and with the help of this capillary tube, we will collect our blood sample. We will slowly insert this capillary tube inside the retroorbital sinus of the mouse, or in case of rat, inside the plexus under the eye portion. For this procedure, we have to use anesthesia to the rodent so that it does not move or create any kind of discomfort during the procedure. Slowly, we have to insert inside the eye of the rodent the capillary tube and collect the blood sample. As we can see, we have collected our blood sample. The procedure says that the retroorbital blood collection is a common technique for obtaining small to moderate blood samples from the rodents. The statement says that it is a blood collection and it is a very common technique to obtain small amount or moderate amount of blood samples from the rodents. The method says a glass capillary or micro hematocrit tube is inserted into the retroorbital sinus of mouse or plexus of rat under the anesthesia. The statement says that we have to use a capillary tube or a microhematocrit tube, which is to be inserted inside the retroorbital sinus or the plexus. The volume that can be collected of the blood sample from this technique is typically 100 to 150 microliter that can be collected from mice and up to 500 microliters from the rats per session. The advantage of this technique is to provide rapid collection with minimal stress when performed correctly by trained personnel. And the ethical considerations that must be followed are institutional animal care guidelines with proper anesthesia and post procedure monitoring. Now we will see the second technique, which is the tail snip blood withdrawal technique. In this technique, again, we will take out one of the animal from the cage and place inside the tubular mouse holder. As we can see, this instrument is the tubular mouse holder, and the tail of the mouse is outside of the tube. With the help of a scalpel, we will make a small incision in the tail for the blood to come out. As we can see, we have placed a small incision, and now with the help of capillary tube, we will collect the blood sample and place it inside a sterile test tube. The procedure of this technique says that Tail snip is a method used to collect small blood samples from the tail tip of the rodent, mainly in young mice and rats. This statement says that this method is used to collect small blood samples from the tail tip of the rodent, and this technique is mainly used for young mice and rats. The method says the tail tip is cleaned and a small segment, 1 to 2 millimeter, is snipped using sterile scissor, followed by a gentle massaging to collect blood. The statement says that the tail tip is cleaned. First of all, we have to clean the tip of the tail, and then we have to make a small segment of about 1 to 2 millimeter, which is snipped with the help of a sterile scissors. And then we have to gently massage to collect the blood. The volume that can be collected of the blood from this technique is 20 to 100 microliter that can be obtained per snip with repeated sampling possible by re-snip after healing. We can uh, continue this procedure for a long time. The advantages of this technique is that it does not require anesthesia and allows serial blood collection in neonatal and juvenile rodents. The ethical considerations requires proper post-procedure care, homeostasis, and adherence to institutional guidelines to minimize the pain and distress for the rodent. The third technique is the tail blood withdrawal technique. In this technique, again, we will take one of the animal, which can be either mice or rat, and again we will place it inside the tubular mouse holder. We will close the end of the holder, and on the other end, the tail will be outside of the holder. Now, in this step, we have to elevate the tail of the mouse, and with the help of the syringe, we have to collect the blood sample. The procedure of this experiment says that tail vein blood collection is a common method for obtaining small, repeated blood samples from the rodents. The statement says that this collection technique is very common and for used for obtaining small amount of bloods. With this technique, we can attain small amount of bloods, but with the help of this technique, we can repeatedly perform this technique for the blood collection. The method is the tail is warmed to dilate veins, and the blood is drawn using a needle, lancet, or capillary tube from the lateral tail vein. The statement says that first we have to warm the tail so that the veins can be dilated, and then with the help of a needle or capillary tube, we will draw the blood out from the lateral tail vein. The volume that can be collected from this technique is 50 to 200 microliter that can be collected per session, depending upon the species of the rodent and the method used to collect. The advantage of this technique is minimal invasive, which allows for multiple collection over time with proper handling. And the ethical considerations are that it requires animal restraint, proper warming, and post-procedure care to minimize stress and discomfort for the rodent. The fourth step, the fourth technique, we have the cardiac blood withdrawal technique. In this technique, again, we will take out our animal and with the help of a syringe, we will withdraw the blood from the heart. In this technique, we will puncture the cardiac region of of the animal and we will withdraw the blood from there. The procedure of this technique says that the cardiac puncture is a terminal blood collection method performed under deep anesthesia or postmortem. The statement means that cardiac puncture, that which means we have to insert the needle inside the heart. This step is known as cardiac puncture. And in this step, blood collection method is performed. And for this step, we have to give deep anesthesia dose to the animal so that it does not feel any pain during the step. The method is a needle is inserted into the heart to the left ventricle or the right atrium to withdraw blood using a syringe, ensuring minimal tissue damage. The volume that can be collected from this technique is large blood volumes, such as zero point 0.5 to 1 milliliter in mice and 3 to 10 milliliter in rats, can be collected, making it suitable for biochemical and hematological studies. The advantage of this technique is that it provides a high yield of blood with minimal contamination from the other tissues. The ethical considerations that are can be performed only as terminal procedure and ensuring humane euthanasia protocols that are followed. And the last technique, the fifth technique, is the posterior vena cava blood withdrawal technique. In this technique, again, we will take out our animal and in the abdominal region or the slight above region of the abdominal, we will insert the needle where the posterior vena cava lies. And with the help of a needle and syringe, we will withdraw blood from the posterior vena cava. The procedure of this experiment says that the posterior vena cava blood collection is a terminal method used to obtain a large, high quality blood sample from rodents. The statement says that posterior vena cava blood collection is a terminal method. And through this technique, we can obtain a large quantity of blood sample, which is of a high quality, which means it does not contain any type of contamination. The method is under deep anesthesia, a needle is inserted into the posterior vena cava, a larger vein near the abdominal organ, to withdraw blood. The volume that can be collected from this technique is large volume, up to 1 milliliter in mice and 10 milliliter in rats, that can be collected and which is suitable for extensive biochemical and hematological analysis. The advantage of this technique is that it provides a high purity blood sample with minimal risk of contamination from other tissue. The ethical considerations are that this is a terminal procedure requiring strict adherence to human euthanasia protocols and ethical guidelines. So now we have discussed all our five different techniques of the blood withdrawal. Let's do a quick recap of these techniques. At first, we have the preparation of these techniques. For that, the equipments required are sterile syringes, needles, collection tubes, antiseptic wipes, and anesthesia. For animal handling, we have to ensure that the animal is calm, properly restrained, and to minimize stress and movement. For that, we have to use appropriate restraint device or manual restraint techniques. Now, the sites for blood withdrawal for rats and mice both are different. For rat, we have the tail vein technique, through which is a very commonly used for small blood volume collection, and and in that the tail is warmed to dilate the vein, and a needle is inserted into the lateral tail vein. Saphenous vein. In this vein is located in the hind leg. This site is used for moderate volume collection, and the leg is shaved. In this step, we have to shave the leg, and then the vein is punctured with the needle. The third is the retroorbital sinus. In this, a large volume of blood collection is done under the anesthesia act. And in this technique, a capillary tube is inserted into the retroorbital sinus, which is behind the eye. For mice, we have the same lateral tail vein technique, the saphenous vein, and in this we have another technique which is submandibular vein. This vein is located under the jaw, and this allows for quick and relatively large volume of collection. And for this, a lancet is used to puncture the vein. The procedure of these techniques are at first the anesthesia, which depends on the volume of the blood required and the site of collection. And anesthesia may be used to minimize the pain and stress. An example is isoflurane or injectable anesthesia are commonly used. For blood collection, there are four steps. The first is the sterilization. We have to clean the collection site and antiseptic wipes to prevent infection. Second, the needle insertion. We have to insert the needle or lancet into the chosen vein with a steady hand. For tail vein collection, ensure the tail is properly immobilized. Third, blood withdrawal. We have to gently draw the required volume of blood into the syringe or collection tube, and we have to avoid excessive suction to prevent vein collapse. And the last is post collection care. We have to apply gently pressure to puncture the site and stop bleeding. And then we have to monitor the animal for any signs of distress or complication. The fourth, we have the volume of blood collection. For rats, it is typically up to 10% of the total blood volume, which can be safely collected at one time. For a 250-gram rat, this is approximately 1.5 to 2.5 ml. For mice, it is up to 10% of the total blood volume that can be collected, and for a 25-gram mouse, this is approximately 0.2 to zero point 0.3 milliliter. The, and the last, post-procedure care. In this, the first is the observation. Monitor the animal for any signs of distress bleeding of or infection to ensure the animal is fully recovered from anesthesia before returning it to its cage. The statement says that we have to ensure that the animal has fully recovered from the anesthesia effect before before we return it to its cage. Then we have the hydration and nutrition. We have to provide fresh water and food to the animal for the recovery. We have to give him him fresh water and high quality food so that it can recover from the procedure. And then we have to consider offering a high calorie supplement if the large volume of blood was collected. If we have collected a large volume of blood, we have to give him high calorie supplement. And at last, record keeping. It is a very important step. We have the document, the procedure, including the volume of blood collected and the site of collection, and any observation about the animal's condition is recorded. Now we have completed our experiment. Thank you for watching this video, and for more such content, follow RJPT Simlab videos. Thank you.